All right, Bismillah. So let's start with section with part 4 of chapter 7. And so far we have talked about enzyme regulation and how enzymes can be controlled. And if you recall from the last slide from part 3, what remains now is the topic of how enzymes do what they do, or what is the active site of enzymes, and that's what we're going to talk about. So we're talking about the active site. And specifically, when we talk about active site, we'll be talking about a few questions that people normally ask, right? So they ask about, how do enzymes do what they do? How do enzymes carry out their mode of action? How do enzymes catalyze reactions? So for this, they have to look at a few questions, and some of the questions that they ask, basically 3 questions. The first one they ask is basically what amino acid residues are in the enzyme active site, right? So there has to be an active site in the enzyme that does the catalysis. What are the amino acids that are there, right? So again, first question is which amino acids are there in the active site. Okay. The second question that people ask is basically what is the relationship of these amino acids with each other in the active site. So if you have, suppose, 4 amino acids in the active site, how are they 4 arranging each other? What is the spatial relationship? Are they like this? Are they like this? Where are they? And then the third question people normally ask is basically what is the mechanism? How exactly are those amino acids in the active site doing what they're doing, right? And so to answer all these 3 questions and to answer this notion of active site, we'll look at chymotrypsin. And chymotrypsin, um, you have to memorize this name. Chymotrypsin is an enzyme of the digestive system, and it is involved in breaking down proteins for food, right? So it digests food, specifically proteins. And the way it digests protein, it breaks down proteins into amino acids, right? And it carries out a selective hydrolysis of the peptide bonds in proteins, in which the carboxyl group of the peptide bond is coming from aromatic amino acids. So what does that mean, right? So suppose you have a peptide bond, so which is C double bond O and H. That's the peptide bond, and then you have C alpha here, and you have C alpha, right? So what chymotrypsin is going to do is going to break down this, right? And it's going to do hydrolysis. It's going to add water to it, right? And at the end, what you should get 2 amino acids. So you have C alpha, C double bond O, and OH. I'm not showing the ionized forms just for the sake of simplicity. And what you should get NH, NH, and C alpha, right? That's what you should get again. Okay. Now what they're saying is, in the case of chymotrypsin will only break down this bond. It'll only break down this bond if this carbonyl group is coming from a group that has an aromatic side chain. So you have to have an aromatic side chain, right? And that's what the point they're trying to make is that the specificity of this bond breakage is dependent on having an aromatic amino acid on this side. Okay, so we're going to study this enzyme called chymotrypsin that breaks only bonds that are coming, the C terminus is coming from an aromatic side chain. Okay, so to study chymotrypsin, what they did, they created a substrate. They created a model substrate. Now this is very common in biochemistry. What they do, they take a substrate that looks like your real substrate. So again, the real substrate would be a peptide bond, so it is not peptide bond, but to the enzyme it looks similar to the peptide bond. Again, peptide bond, we said, it has to have a C double bond O N H, that kind of thing, right? So you have a carboxy group here that looks that can look like a substrate. So this carboxy group here is also here. So to enzyme it looks similar, and this is the enzyme. And what they found that if you take this substrate, which is called para-nitrophenyl acetate, a model substrate, you put the enzyme in it, and what the enzyme does, it breaks down right here. It cuts it at thinking that this is a peptide bond, and it gives you one half, which is this pair of nitrophenolate, which is this half. This whole thing comes off. Oops. Um, this half of the molecule comes off, and then the second half comes out here, which is the second half, right? So it breaks into two pieces. Now, when they're doing it, this experiment, what they found that this is actually, it happens in two different steps. It's a step one, step two. It is not like, if you have, suppose you have a pencil, right? And then if you're trying to break it, you just break and then you get two products out, two halves out. They didn't see that. What they found that it is in step one and step two. They have first this thing comes out and then later on this thing comes out in two discrete steps. So that was the first clue of how it may be working. And this is shown here that para-nitrophenolate acetate, the model substrate, is hydrolyzed by chymotrypsin in two stages, right? And what they found in first stage, you get one half of the product, and then later on you get the second half of the product, right? So there are two stages. At the end of stage one, the para-nitrophenolate ion is released, and at the end of stage two, the second part is released, right? So please look at the book. The book will explain again what is meant by this, and we'll talk more about this as the lecture goes on. Okay, so that's the first thing that they found out, that the enzyme actually has two-stepped reaction. Okay, and then they wanted to look at what amino acids are involved in the active site, and what they did, they used some chemicals, and this is one of them. It's called DIPF. Um, I don't want you to memorize this. There's no need for that. It's a chemical that reacts with serines, right? Specifically, it reacts with serines. And when they use this chemical, they found that chymotrypsin dies. It's not active anymore. So that means that this guy is reacting with serine and inactivating the enzyme. Therefore, chymotrypsin is a protease that needs serine. So we call it a serine protease, right? So first hint was that you need to have a serine in the active site. So that's one amino acid. And then they did some more experiments. They used a different compound called TPCK. This is again another compound that reacts specifically with histidines, right? With histidines. And what they found that this is not showing. It's not showing. Yes, yeah, histidine. So what they found that if you add TPCK, then chymotrypsin becomes inactive again. That means chymotrypsin needs histidine as well. So you have now serine that it needs, okay, and now you found it needs histidine as well. So that's really interesting. So it has those two amino acid side chains. Okay, so they found serine at 195, which is important, and they found histidine at 57 that are important. And then they purified the enzyme, and they got the whole structure of chymotrypsin, and using X-ray crystallography, they look at the amino acid in the active site. They found, sure enough, in the active site you have histidine, you have the serine, and they also found this one, aspartate 102 as well, the third amino acid. And you'll see that this is also important as well. And if you look at the structure, and this is the structure shown here, this is the whole chymotrypsin molecule. This is the active site inside it. And if you look at the active site, you find that you have serine, as we thought you would have. You have histidine, and then you have aspartate. And then you find that those three amino acids are very close to each other, and they are almost in a straight line. So it almost looked like it's like a straight line, right? And that was really the interesting thing that they found about this thing. Okay, so histidine is important. Serine is important. This is serine. Histidine is important, and then they found that aspartate is important as well. Okay, now we'll talk about, and this is called catalytic triad, like those three amino acids that are catalytic, right? So these are three really interesting links that are in here that I want you guys to look at, and you'll find that these are extremely useful, all three of them, and we'll go one by one. So this is the first one, link here, and what they're showing you that, again, this is a peptide bond that serine cuts, chymotrypsin cuts, right? But chymotrypsin cuts cleavage of polypeptides that are adjacent to an aromatic amino acid. Remember? So that's where it cuts. Okay. It turns out that there's another. So this is chymotrypsin. So let me just highlight in yellow. So this is chymotrypsin. Okay. There's another protease called trypsin, right? So trypsin is a protease that also cleaves at the peptide bond, right? But it says it wants a peptide bond in which the carboxy carbonyl is coming from an amino acid which is not hydrophobic, rather which has a positive charge. So this is a different amino acid, and here you have aromatic amino acid, right? And that's the big difference between the two enzymes. So, and this actually drives the substrate, drives the specificity, meaning the active sites of trypsin and chymotrypsin must be different to accommodate these two substrates, right? So if you click on this link, you'll find very nice information, and we'll see that later on as well. So in fact, it turns out there are other proteases, not just trypsin and chymotrypsin. So there's chymotrypsin, there's trypsin, there's thrombin and protease and cetylase and carboxypeptidase, and they all have different specificities, meaning, they all will cut here. Yes, they all will cut here, but some depend on what is here and some depend on what is on this side, right? So as we said, trypsin wants positive charge amino acids, so trypsin wants lysine and arginine. Chymotrypsin wants aromatic here, right? And then you have all the other ones. So I just want you to know these two. I want you to know trypsin, and I want you to know chymotrypsin as those amino acids. Those proteases. Okay. Again, if you have chymotrypsin as the enzyme, so this is the enzyme binding pocket, then the substrate, which is shown in here, so which I'm gonna put light in green color, this is substrate. So the substrate would actually have a bulky or aromatic side chain. So you can see how it fits very nicely. In the case of trypsin, as I said, trypsin, again, this is the binding pocket, but trypsin binds to arginine. It binds to arginine, for example, as a substrate. That arginine has a positive charge. That means you must have a negative charge here. You must have a negative charge here to be able to bind to the lysine in here, right? And in this case, you must have a big pocket, a hydrophobic pocket in the case of chymotrypsin, right? Likewise, if you have elastase as a third different protease, for example, elastase can bind to substrates that are small and uncharged. That means the binding pocket should be really small, should not be really big, and really has a very small binding pocket, right? As opposed to trypsin. So this concept of specificity is very important, and you should know the difference between trypsin and chymotrypsin specificity. So chymotrypsin binds substrates that have this aromatic or hydrophobic ring, right? Okay, but trypsin, on the other hand, binds to substrate that actually have a positive charge group like arginine and lysine.
All right,
So let's see if this
I'm going to be looking at one of the links that I
actually
sent you, that I
works.
just
No, it's
listed
not
in
working.
the previous slide.
Let's
And
see.
this
Uh,
website shows very
okay,
nicely
and I think
the
I'll
concepts
send this to
of
you as a
enzyme
separate file. So see if you
specificity,
can figure this out
how
and
enzymes can
play
bind
this.
to
I think it should work
specific
as a
substrates,
separate file.
as well as the
Okay, so I'll
binding model
stop
that
with
we've
this
talked
for the
about
time
earlier,
being.
the lock-in key versus the induced fit model. Right? So I'm expecting all of you to please read whatever is here on the left-hand side. It's really important. Now, what's showing here is a picture of chymotrypsin, the enzyme that we're looking at, and it's got a substrate bound in it, and they're highlighting a few of the enzymes that are involved in catalysis and the enzymatic reaction. So you have the histidine and aspartate, and you have the serine here somewhere. Okay, so after navigation through this section, you will have a clear idea of various factors that influence substrate binding, as well as current models of enzyme and substrate binding. Okay, so again, this is showing you the binding pocket of chymotrypsin and the three amino acids that we know are critical, which are aspartate, shown in green here, histidine in purple, and serine shown in this orange yellow color, right? And what they're showing you also this hydrophobic pocket in blue, and showing you in white is the peptide substrate, which is in the active site, and you can see the aromatic ring here, which is very nicely um, uh, resting in the hydrophobic pocket, and that's what's driving the substrate. So the text says the behavior of an enzyme has its physical basis in the three-dimensional structure of the active site, which is part of the protein that interacts with the substrate. And then go down. So what they're saying is complementary fit between the substrate and the enzyme accounts for the enzyme specificity. And this notion of complementary fit, it's just like a lock and key, right? So that determines the enzyme specificity. If a key cannot fit in the lock, then that substrate cannot be acted upon by the enzyme. So as an example, we know the chymotrypsin catalyzes the same reaction as other proteases like trypsin. So trypsin and chymotrypsin and there are many other proteases. They all catalyze the same reaction, which is breaking this peptide bond. This is C double bond O NH peptide bond, C double bond O NH peptide bond. However, what they're saying is but chymotrypsin acts or recognizes only peptide bonds next to aromatic amino acids. So if you have an aromatic amino acid at the C terminal of those peptide bond, that's what chymotrypsin cuts in. So this is again a peptide bond, but this is not aromatic. So chymotrypsin will not work on this, but it will work on this. So that's what the text says. Chymotrypsin catalyzes the cleavage of polypeptide chains adjacent to the C terminal side chain of aromatic amino acids. This, if you have a positively charged amino acid, this is what trypsin works on. So trypsin does not work on hydrophobic, but trypsin works on a positively charged amino acid like lysine or arginine, and chymotrypsin has this aromatic specificity. Okay, so click on the amino acid with the aromatic side chain. So this is not aromatic, this is not aromatic, and this is aromatic. Okay, now what's shown here is this blue. They're showing you, a typical given hypothetical enzyme that has amino acid side chains, which are shown in this alanine and isoleucine, and then these are aspartate and amino and water and another amino group here. So these are all a protein, but what's shown in the substrate is in red. So this red guy is a substrate, right? So they're showing you a substrate bound to a binding pocket in the protein. And what they're saying on the left hand side, that non-covalent interactions between substrate, which is the red guy here, and the active site, which are all the amino acids that are shown here, side chains are shown here, contributes to the affinity and specificity of the binding between the enzyme and substrate. Examples of these interactions that we know are hydrogen bonding and ionic charge interaction, as well as hydrophobic interactions. Right? And these interactions are what gives you the initial binding of the substrate. Right? So they want you to, so if you have this is a charge interaction, this will be hydrogen bonding between the substrate and the active site. This is again a charge interaction, and this would be the hydrophobic interaction because you have non-polar groups. Okay, now they're saying you what do the amino acid side chains next to the trypsin cleavage site. Now again they're showing you this is a trypsin substrate, this is a trypsin substrate, meaning this is a substrate that trypsin can work on, can cleave here. This is a trypsin substrate that can cut here, and they're asking you what is common, what is this and what is this. So these are both, they're not aromatic, they're not hydrophobic, and they're not acidic. Acidic, remember, will have COOH. So this is the basic, they have basic side chains, right? Not acidic. So this is basic. Okay, now given that the substrate residues are basic and positively charged, which complementary amino acid would you expect trypsin to have in the active site next to the substrate? So if the substrates are positive, right, then the amino acid side chains that are interacting with them must be the opposite, which are acidic. Okay, and that's what they're showing you. This is trypsin. In trypsin, they're showing you the substrate. This is the substrate that has an amino charged substrate, and the active site will have a negatively charged amino acid in trypsin, which is the carboxylate amino acid. That's what they're talking about in the previous slide. Here, chymotrypsin has a hydrophobic binding pocket. That's where the substrate with the hydrophobic side chain will fit in there. Okay, I think we'll just stop here for now. Let's see. Okay, so this is the active site of a serine protease like chymotrypsin, right? So this is shown in blue. This is what the protein looks like, and you can they're showing you a hydrophobic pocket. And it's also showing you things that you should keep in mind. The serine at 195 is showing you a histidine at 57 right next to it. It's also showing you an aspartate right next to the histidine. So we're going to add the substrate, and the substrate comes in, and notice the substrate has a hydrophobic aromatic ring. That's where it fits into the hydrophobic pocket of the protein of the enzyme, right? And then also it positions the peptide bond, the serial bond ONH, right next to the serine and ready for the attack. And so when you start the reaction, the electrons take the proton from the serine, and the electron attack the carbon here, and the electrons double bond is moves over there, and you break this bond and you form a covalent bond with serine and the substrate. If you continue, the electrons, it resonates, comes back here, and your bond is broken, and the proton is taken up by the nitrogen. The nitrogen takes a proton, yeah, it comes off, right? And then water comes in. Again, the same reaction. This is a partial positive carbon, so this electrons comes here, takes over there, you attack that, it goes over there. And then the last step is basically a reversal of the second step, and you have the second half of the substrate being released now, and then the enzyme returns in its original position. Okay, so let's see if we can stop this.